will begin by talking about the structure and function of a neuron. Neurons are the nerve cells that transfer information throughout the body. They use both long-distance electrical signals and short-distance chemical signals for communication. Here pictured is the basic structure of a neuron. First, most of the neuron's organelles, including the nucleus, are contained in the cell body. Also shown are the dendrites, which are multiple branched extensions which are able to receive multiple cell signals from other neurons. The stimulus is received by these dendrites of the presynaptic cell, and the signal then travels to its axon. The axon transmits signals to other cells. It is covered by myelin sheath, which functions as insulation for the signal to travel faster and more efficiently, similar to the way rubber insulates a wire. At the end of the axon, there is a junction called the synapse, where the signal is transferred from the synaptic terminal of the presynaptic cell, or the branches at the end of the axon, to the postsynaptic cell. The signal then continues in this direction.
All cells have membrane potential, meaning they have a voltage across their plasma membrane. When neurons are not sending signals, they have a resting potential between -60 to -80 millivolts. This potential is achieved through the ions within and surrounding neurons. The inside of the cell has a high potassium and anion concentration, making it negatively charged. However, the outside of the cell has a positive charge with a high concentration of sodium and chloride. There are sodium-potassium pumps in the plasma membrane that use ATP hydrolysis to actively transport sodium out of the cell and potassium inside the cell. The concentration gradients of both sodium and potassium create a chemical form of potential energy. There are also ion channels along the membrane that have selective permeability, allowing only potassium or sodium to flow through. Potassium ions can flow out of the cell through certain channels, but the anions always stay inside. Sodium ions can flow inside the cell, making the inside less negative. However, the neuron's resting potential is negative because there are more potassium channels than sodium channels. Changes in the membrane potential and permeability result because of gated ion channels, which are mostly voltage gated. As these channels open or close in response to stimuli, the membrane potential will either hyperpolarize or depolarize because of the movement of ions. Once the depolarization hits a certain value called the threshold, which for mammals is -55 millivolts, the action potential is triggered. Action potentials are nerve impulses that carry information along an axon to another neuron or receiving cell. Once the nerve cell is stimulated, the sodium channels open up and the sodium ions diffuse inside the cell, as seen in the second picture. The wave of nerve impulses travels down the neuron, creating the action potential. Then, as seen in the third picture, potassium channels open up and the potassium ions flow outside the cell. In order to reset, the sodium-potassium pump uses 1 ATP to push 3 sodium ions out and 2 potassium ions back in, and this process is repeated until the resting potential is resumed. This period is known as the refractory period, in which the action potential cannot be initiated right after the first action potential, as the sodium channels remain inactivated.
This is a graph showing action potential occurring inside a neuron. At the first point, the neuron is at resting potential. At the second point, the sodium channel opens, allowing sodium to flow out of the neuron, which depolarizes the neuron and increases the membrane potential. The membrane potential increases until it hits the threshold, causing action potentials to occur. At the third point, the rest of the sodium channels open due to the membrane potential of the neuron hitting the threshold. At the fourth point, the membrane potential reaches hypershoot, which is the highest point the neuron can go to. The sodium channels close at this point, and the potassium channels open, allowing potassium to flow out of the neuron. This can be seen at the fifth point, where hyperpolarization occurs. The membrane potential decreases until it hits the undershoot, which closes the potassium channel slowly. At the sixth point, the sodium-potassium pump sodium and potassium back into their respective places, meaning sodium outside and potassium inside, which puts the neuron back into its resting state.
Now let's talk about how neurons communicate with other cells and how the information is passed along. The picture that you see on your screen is a diagram of this process. So to start off, The plasma membrane of the synaptic terminal opens calcium channels when the action potential depolarizes. The calcium channel is voltage gated, and it allows for calcium to enter the terminal. The high concentration of calcium in the terminal results in the synaptic vesicles to fuse with the presynaptic membrane, and this releases the neurotransmitters into the synaptic cleft. As the neurotransmitter is released, it changes from an electrical to a chemical signal. The neurotransmitters then bind to the receptor on the ligand-gated channels, which causes it to open up and for the message to be transferred. This allows for the neurotransmitter to change back from a chemical to an electrical signal. When it changes back to an electrical signal, it can either be inhibitory, which hyperpolarizes the postsynaptic neuron, or excitatory, which depolarizes it. Depending on which type of neurotransmitter is greater in amount, there may or may not be an action potential. However, after the message has been transferred, the neurotransmitters release from the receptor and either degrade or get recycled. This is an explanation of the process of how neurons communicate with other cells at the synapses. Thank you for watching.